Hey there, listeners. Welcome to another paper preview podcast. Credit for this work goes to the original authors. Please find a link to their paper on PubMed Central in the description below or using the QR code on screen. This paper preview is meant to be a general overview and introduction to the paper. Please read the actual text and figures for more details. The following discussion, script, and voices were created using AI and are not intended to mimic the likeness of the original authors. As with any use of AI, all factual information should be double-checked with the original source.
Welcome back to the Deep Dive, where we plunge into the most groundbreaking research to extract those aha moments and make you incredibly well-informed. Today, we're exploring a fundamental question in cell biology, one that truly pushes our understanding of cellular resilience and adaptation. We're going to dive into the incredible efficiency of a cell, this miniature factory, constantly building and rebuilding its molecular machinery with breathtaking precision. Think about the ribosome. It's not just another cellular component, it's a massive resource-intensive complex, absolutely essential for every living cell, responsible for synthesizing all the proteins that make life possible. But here's the fundamental challenge that this paper addresses. What happens when cells are suddenly slammed by stress, like a sudden rise in temperature, and can't build these crucial ribosomes at their usual breakneck pace? Okay, let's unpack this. Imagine a cellular crisis, like that heat shock. Ribosome production, which is usually a finely tuned symphony, grinds to a halt. But for a time, the cell might still be making the individual building blocks, the ribosomal proteins. What happens to these orphan proteins, the ones that don't find their RNA partners? Are they just wasted, degraded, perhaps forming toxic aggregates? Or is there a more sophisticated adaptive strategy at play? Today, we're diving into a fascinating paper that unravels precisely this problem. As an editor at Nature Cell Biology, I can tell you this research immediately stood out for its elegance and the profound implications of its findings.
Thank you. It's truly exciting to discuss this work. This deep dive is based on our paper titled Adaptive Preservation of Orphan Ribosomal Proteins in Chaperone-Dispersed Condensates, published, as you mentioned, in Nature Cell Biology. It was a truly collaborative effort, bringing together multidisciplinary approaches from a fantastic team of researchers at the University of Chicago. We were really trying to answer a core question about cellular resource management under duress. How does the cell cope?
And for you, our curious graduate students in biomedical research, this deep dive is particularly relevant. This paper isn't just about a specific mechanism. It's a masterclass in combining cutting-edge methodologies to unravel a complex biological problem. It offers profound insights into protein homeostasis, the intricate dance of cellular stress responses, and the dynamic nature of cellular compartments, specifically biomolecular condensates. Our mission today is to extract the most important nuggets of knowledge from this groundbreaking research, giving you a shortcut to understanding the complexities of cellular resilience and perhaps sparking new ideas for your own research. So let's begin by really appreciating the sheer scale of ribosome biogenesis, or RIBI, because it absolutely sets the stage for understanding the orphan problem. This isn't just any cellular process. It's one of the most resource-intensive activities a cell undertakes. From an editor's perspective, the numbers alone are eye-opening.
They really are. If we consider a rapidly dividing cell, like yeast under ideal nutrient-rich conditions, the production of ribosomal proteins, which we'll refer to as RPs, can account for up to half of all newly synthesized proteins in the cell.
Half. Wow.
Yes, half. We're talking about an enormous ongoing investment of energy and resources. To put that into perspective, in a single yeast cell, over 100,000 RPs are synthesized every minute.
Every minute.
Every single minute. That's a staggering production line running continuously.
100,000 a minute. That's a number that truly emphasizes the cell's commitment to growth. And it also means that RIBI is often the rate-limiting step for cell proliferation. If a cell wants to grow and divide efficiently, it absolutely needs to make ribosomes, and a lot of them, very quickly. So this massive constant investment really underscores why the efficient management of these RPs is not just important, but absolutely crucial for the cell's overall fitness and survival. You simply can't afford to be wasteful with such a core, high-volume component.
Exactly. And that brings us directly to the orphan problem. A complete ribosome is a finely tuned, sophisticated machine composed of 79 different ribosomal proteins and 4 ribosomal RNA molecules, or rRNAs. These components don't just randomly assemble, they need to come together in a very precise stoichiometric manner. The inherent challenge is that many RPs are intrinsically aggregation prone. They don't like to be alone. In the absence of their RNA partners, which act as a structural scaffold, they tend to misfold and clump together, forming potentially toxic aggregates.
So they're like very sticky puzzle pieces that only make sense and stay well-behaved when they're together.
Yeah.
Otherwise, they become a problem.
That's a great analogy. And this challenge, as you described, is significantly compounded by cellular compartmentalization. Indeed, the RNA, the other major component, is transcribed in a specialized compartment within the nucleus called the nucleolus. However, the ribosomal proteins themselves are synthesized out in the cytosol on other ribosomes. So you have these two crucial components being made in different cellular locations, and the RPs then need to be actively transported from the cytosol into the nucleolus by a specific family of transport proteins called karyopherins and their cofactors, escortins, to meet their RNA partners and begin assembly. It's a complex logistical operation, even under normal conditions.
That's already a significant coordination challenge. But then layer on top of that a stress response like heat shock, which is a common cellular perturbation. What happens to this delicate, highly coordinated process during such a crisis?
Right. During stress, whether it's heat shock, nutrient limitation, or other environmental insults, cells have a very rapid and powerful response. They quickly repress RNA transcription. For instance, in yeast, even a moderate temperature increase to 37 degrees C can trigger this repression. However, and this is a critical point of mismatch, protein synthesis, including the continued synthesis of RPs, can carry on at significant rates, especially at more moderate heat shock temperatures like 39 degrees C.
Okay.
Complete repression of all protein synthesis usually only occurs at more extreme temperatures, typically above 40 degrees C in yeast.
So if I'm understanding this correctly, the cell effectively stops making the RNA scaffold, but it's still churning out the RP building blocks, at least for a while, creating this imbalance.
Precisely. This mismatch leads to a massive and rapid buildup of what the researchers in this paper call orphan ribosomal proteins, or ORPs. These are RPs that haven't bound to RNA, or perhaps they've been mislocalized, or are simply in excess. They're essentially surplus parts floating around, and given their aggregation-prone nature, as we discussed, they pose a serious proteotoxic threat to the cell if left unchecked.
So before this work, what did we understand about how a cell typically deals with these potentially dangerous ORPs? We know cells have very robust quality control mechanisms.
They absolutely do, and these are well described. Cells have several mechanisms for dealing with ORPs, primarily through degradation pathways. These are often mediated by specific ubiquitin ligases, such as Tom1-Shu-W1 in mammals and UBE2O, which essentially tag the ORPs with ubiquitin, marking them for destruction by the proteasome.
The standard cleanup crew.
Exactly. This is a very efficient way to eliminate misfolded or surplus proteins. In fact, we know that imbalances caused by loss-of-function mutations in RP genes are known to cause a class of human diseases called ribosomopathies, which include conditions like Diamond-Blackfan anemia.
Which really highlights just how critical this stoichiometric balance of ribosomal components is for cellular health, and how quickly its disruption can lead to serious pathology.
And further, the accumulation of ORPs is known to activate a broader ribosome assembly stress response. This response does several things. It transcriptionally downregulates the genes encoding RPs, trying to reduce the supply.
Okay, turn down the tap.
And it also induces the heat shock response, or HSR, which is a transcriptional regulon that produces more protein homeostasis factors, like chaperones, to help deal with the overall proteotoxic burden. This tells us that ORPs are under constant surveillance by the proteostasis network. The cell knows they're a problem and actively tries to mitigate their damaging effects.
So the cell knows ORPs are a problem, and it has these robust mechanisms to degrade them. But this paper takes us in a different, perhaps more adaptive and resource-efficient direction. Given these established degradation pathways, what made your team suspect there was a more sophisticated, adaptive mechanism at play beyond just destruction? What was the initial hint that something else might be going on?
That's a very perceptive question, and the initial hint actually came from observing the behavior of a specific chaperone protein called cis1 in yeast and its human homologue, DNA-JD6. These are J-domain proteins, or JDPs, which are crucial co-chaperones that regulate the activity of the major HSP70 chaperone system. Researchers already knew that CIS-1 relocalizes dramatically during heat shock, and that this relocalization is one of the earliest known events in the proteostasis network's response to stress. It happens incredibly rapidly, suggesting it might be an immediate cellular triage event rather than a cleanup operation.
So the cell's initial alarm system, this CIS-1 protein, quickly moves somewhere specific when heat stress hits. Where exactly did it go, and how did you pinpoint its new location with such precision?
To pinpoint that, we employed a sophisticated experimental setup, a 4-color yeast strain. This allowed us to monitor CIS-1 Mervinus localization in live cells relative to several key cellular landmarks simultaneously. We used NSR1 Mer-scarlet I to mark the nucleolus, Sec61 halotag JF646 for the nuclear boundary and cortical endoplasmic reticulum, and HSP104-MELA-TFP1 for cytosolic aggregates. Crucially, we use lattice light sheet microscopy, which offers exceptionally high spatiotemporal resolution, allowing us to capture these dynamic changes in 3 dimensions over a very short time course during heat shock.
4 colors and lattice light sheet. Okay, state-of-the-art imaging. And what did this advanced imaging reveal? What was that rapid relocalization like?
Under non-stress conditions, CIS-1 was generally found diffused throughout the nucleoplasm and cytosol, pretty spread out. However, upon acute heat shock at a moderate temperature of 39 degrees C, there was a very rapid and striking relocalization event. CIS-1 moved distinctly from the nucleoplasm to a specific region surrounding the nucleolus, what we termed the perinucleolar region, and this occurred within a remarkably fast 2.5 minutes.
Wow, 2.5 minutes.
Yeah. It wasn't just moving, it was congregating at a highly specific strategic locale.
That's incredibly quick for cellular response, suggesting it's a very early and intentional action.
Exactly. Image analysis confirmed this increased CIS-1 perinucleolar localization in almost all cells. Interestingly, we also observed the formation of cytosolic foci containing both CIS-1 and HSP104, which are classic stress granules. But these formed with slightly delayed kinetics compared to the perinucleolar localization, suggesting distinct roles or triggering mechanisms. Most importantly, this rapid CIS-1 relocalization to the nucleolar periphery was completely abolished if cells were pre-treated with cyclohexamide or CHX, which is a potent inhibitor of protein synthesis.
Ah, the CHX sensitivity again.
Yes. This indicated that ongoing protein synthesis is absolutely required for CIS-1's dynamic movement and accumulation during heat shock. It wasn't just moving to a pre-existing structure, something new had to be made.
Here's where it gets really interesting, because that dependency on ongoing protein synthesis is a critical first clue. With CIS-1 rapidly relocalizing to a specific site, dependent on newly made proteins, the next critical question, of course, was who was it moving with? How did you begin to unravel its new network of interactions in this perinucleolar location?
That was indeed the crucial next step. To identify CIS-1's partners, we employed a powerful, multi-pronged proteomic approach. First, we used a technique called proximity-dependent labeling coupled with mass spectrometry, or APEX-2MS.
Okay, APEX-2MS. For our graduate student listeners, could you walk us through the core principle of that and perhaps its advantages compared to traditional immunoprecipitation for probing these kinds of dynamic interactions?
Certainly. In APEX-2MS, we genetically fuse CIS-1 to an engineered peroxidase enzyme called APX2. When this enzyme is activated by hydrogen peroxide, it generates highly reactive biotin phenoxyl radicals that covalently attach biotin tags to proteins that are in very close proximity, typically within a few nanometers, to the CIS-1 APEX2 fusion protein.
Like a little labeling cloud around CIS-1.
Exactly. After heat shocking the cells and performing this proximity labeling, we can then purify the biotinylated proteins and use mass spectrometry to identify them. The major advantage over traditional IPMS is that APEX-2MS can capture transient or weak interactions, as well as proteins that are simply nearby in a specific microenvironment, even if they're not tightly bound.
Got it. So it's better for things that might just be, passing through or part of a dynamic structure.
Precisely. This is particularly useful for dynamic cellular compartments or rapidly forming complexes where stable binding might not occur.
And what did that very broad, unbiased survey tell you about CIS-1's immediate neighborhood during heat shock?
The APEX-2MS results were quite revealing. We identified 698 proteins that were reproducibly labeled. CIS-1 was found to be proximal to other chaperones, various metabolic enzymes, and factors involved in translation and ribosome biogenesis. But the most prominent and enriched class of proteins were ribosomal proteins, or RPs, by far. Furthermore, we noticed that cis-1 interactors were significantly depleted for intrinsically disordered regions, or IDRs, compared to the whole proteome, suggesting that cis-1 was interacting with more structured protein domains rather than just amorphous, highly disordered regions.
That's a strong indicator that cis-1 is specifically engaging with RPs, and perhaps in a more structured way than simply cleaning up random misfolded proteins. How did you then build upon those findings using the second proteomic approach?
Right. To complement the Apix-2 method, which captures proteins in close proximity, we performed immunoprecipitation, IPMS, of cis-1, again after heat shock, but with a crucial twist. We compared conditions with and without cyclohexamide, or CHX, a protein synthesis inhibitor.
The CHX comparison again. Smart.
This allowed us to specifically identify interactors whose association with cis-1 was dependent on ongoing protein synthesis. The rationale here was that if cis-1 was interacting with newly synthesized RPs, then blocking protein synthesis with CHX should eliminate, or at least significantly reduce, that interaction.
So any interaction that significantly disappeared when protein synthesis was blocked would likely be with newly made orphan proteins. That's a very clever control.
Precisely. Out of 247 reproducible cis-1 interactors, a remarkable 209 were significantly depleted in the CHX-treated cells.
209. That's most of them.
Yes, the vast majority. This strongly confirmed their dependence on ongoing protein synthesis. This category included other chaperones like HSP70 and HSP104, whose interactions with cis-1 actually increased in CHX-treated cells, suggesting they might be dealing with existing, more stable aggregates when new synthesis is shut off.
Interesting nuance there.
It also included co-translational folding factors and degradation factors. But strikingly, the most abundant class of CHX-sensitive cis-1 interactors was, once again, ribosomal proteins.
That's a powerful convergence of data. Two different methods, Apix-2 proximity and CHX-sensitive IP, both pointing overwhelmingly to RPs as key partners for cis-1 during heat shock, and specifically, newly synthesized RPs. That's difficult to argue with.
Indeed. When we looked at the intersection of both the Apix-2 and the CHX-sensitive IP lists, we found 178 common proteins, and a truly remarkable 49 of these were ribosomal proteins.
49 RPs common to both.
49. Gene ontology analysis overwhelmingly confirmed the strong enrichment for RPs. What's fascinating here is that cis-1 isn't just interacting with any RPs. These are specifically the newly synthesized ones, suggesting a proactive role in managing new production rather than just cleaning up mature ribosomes or general cellular clutter.
That distinction between newly synthesized and mature RPs is absolutely critical for understanding the mechanism. How did you definitively prove that cis-1 was interacting with orphan RPs, that is, RPs not yet incorporated into ribosomes, rather than simply engaging with existing, fully formed ribosomes?
To definitively answer that, we devised an elegant in vivo pulse chase labeling strategy using the halo tag system. We endogenously tagged several RPs from both the large and small ribosomal subunits with halo tag. This allowed us to selectively label either mature, pre-existing RPs or newly synthesized RPs, effectively giving us a temporal handle on their fate.
So you could literally track the destiny of old versus new proteins in real time, like painting them different colors.
Exactly, that's a good way to put it. For new RPs, we first masked all mature pre-existing halo tag RPs with a non-fluorescent blocker. Then we added a fluorescent dye to label only the RPs made after that block.
Got it.
For mature RPs, we did the opposite label first, then chase. When we performed immunoprecipitation of cis-1 using lysates from these labeled cells, we found that cis-1 co-precipitated only with new RPs and only during heat shock.
Only the new ones and only under stress.
Correct. Mature RPs, on the other hand, showed no interaction with cis-1. This was a clear and direct indication that cis-1 was specifically engaging with the newly synthesized unincorporated proteins.
And what did live imaging in these cells reveal about their localization in relation to cis-1? Did it match the biochemistry?
The live imaging corroborated the IP results beautifully. Under heat shock for 10 minutes, mature RPs did not form perinucleolar structures or cytosolic foci. They stayed put, essentially. However, newly synthesized RPs, specifically those like RPL26A, RPL25, RPS4B, and RPS9A, which are all known to be early assembling RPs that incorporate into ribosomal subunits in the nucleolus, accumulated dramatically at the nucleolar periphery. And crucially, these newly synthesized ORPs co-localized extensively with cis-1.
So these specific types of ORPs, the ones destined for the nucleolus, are the ones cis-1 is keeping an eye on, right at that vital nucleolar boundary. This is a highly specific surveillance system.
Precisely. To further confirm this specificity, we also tested RPs that are known to join the ribosome at a later stage in biogenesis, typically in the cytosol, such as RPL29 and RPS3. These late-joining RPs did not localize to the nuclear periphery or co-localize with cis1.
Okay, reinforcing the idea it's about the early nuclear-bound ones.
Yes, this strongly confirmed that cis1's engagement was specific for nucleolus-bound ORPs, those orphaned, at the very site where their RNA partners would normally be synthesized. The system is remarkably selective in its target.
And critically for broader implications, did you extend these fascinating findings to human cells? Is this a conserved mechanism?
We did, and it is conserved. Using HCT116 human cells stably expressing RPL26 halo, we performed similar pulse chase and heat shock experiments. We found that newly synthesized RPL26, highly concentrated at the nucleolar periphery and co-localized extensively with DNJB6, the human homolog of cis1, during heat shock at 43 degrees C for 30 minutes. This mirrored the yeast findings almost perfectly, indicating a fundamental and conserved mechanism across species.
Fantastic. And were these ORPs at the nucleolar periphery truly isolated, or were they still associated with other ribosome biogenesis factors? What does this tell us about the exact subcompartment and the definition of their orphan state?
That's a good question. To investigate this, we tagged 15 different factors involved in 60S ribosome biogenesis. We found that indeed early nuclear assembly factors like RPF1, YTM1, NUP16, TFIX, and NOG1 did co-localize with RPL26A at the nucleolar periphery.
So not completely alone.
Not completely alone, suggesting a loose association with an early stage of the assembly pathway. However, we observed no co-localization with rRNA biogenesis factors like RPL190 or NSR1, nor with most nucleoplasmic or cytosolic assembly factors. This implies that while these ORPs are in a specific subcompartment, they are effectively orphaned from their crucial RNA partners and from progressing further in assembly, essentially being held in a paused state.
Okay, so paused early on, near the start line, but not able to run. This multi-pronged approach from proximity labeling to sophisticated live cell imaging across species really solidifies the idea that cis1 and its human counterpart are specifically engaging with these newly made, unribosome-bound ribosomal proteins under stress right at the nucleolar periphery. It paints a vivid picture of the cellular triage system. But how do we know it's the ORPs themselves driving cis1's recruitment, and not just cis1 moving there independently for some other reason? Establishing causality is key for a paper of this caliber.
That's a crucial point, and we went to great lengths to establish that causal link. In yeast, we use an auxin-inducible degradation system to rapidly and acutely deplete IF1.
IF1. Remind us what that does.
Right. IF1 is a transcription factor that's absolutely essential for RP gene expression. By depleting IFF1, we could effectively turn off the faucet for new RP production.
So by removing IF1, you could essentially ask, if there are no new ORPs, does cis1 still go to the nucleolar periphery?
Exactly. That was the experiment. When these IF1 depleted cells were subjected to heat shock, we observed a reduced transcriptional induction of HSF1 regulated genes, which are part of the broader heat shock response. This suggested that ORPs themselves contribute to the activation of the HSR, acting as a proteotoxic signal.
Interesting side point there, the ORPs as HSR triggers.
Yes, but most critically for cis1 localization, the rapid loss of IF1 nearly abolished cis1 localization to the nucleolar periphery during heat shock in most cells.
Nearly abolish. There's powerful direct evidence. No ORPs, no cis1 at the nucleolar periphery. That really underscores the ORPs as the active signaling component.
It is. However, it's worth noting that if F1 depletion did not prevent the formation of cis1 cytosolic foci, indicating a specific effect on the perinuclear cis1 localization rather than a general chaperone defect. We further validated this in human cells. Using the mTOR inhibitor taurin-1, which effectively reduces RPL26 expression, we found that this treatment prevented DNAJB6, the human cis1 homolog, from localizing to the nucleolar periphery during heat shock.
Consistent cross-species again.
Yes, providing further evidence for a conserved causal link. So yes, it's not just that cis1 is there when ORPs accumulate. The ORPs themselves are actively signaling for cis1's recruitment, highlighting their importance in orchestrating the stress response.
So we have ORPs accumulating, and they're actively calling in cis1 to the nucleolar periphery. What exactly are these ORPs doing there, and what is cis1's role once it arrives? The paper describes them forming dynamic, chaperone-dispersed condensates. That term alone is quite descriptive, but what does it mean in practice?
This is where the visualization of cellular dynamics really shines, and it was a pivotal discovery. Using 4D lattice light sheet imaging, which allows us to perform time-lapse imaging in 3 dimensions with incredible speed and detail.
The 4D again, yeah.
We observed newly synthesized RPL26A and Cis1 during a sustained 10-minute heat shock. What we saw was truly remarkable. RPL26A and Cis1 remained localized at the nucleolar periphery in dynamic clusters that rapidly reorganized through processes of fission and fusion. We term these structures ORP condensates.
Fission and fusion sounds quite liquid-like, as if they're constantly flowing and merging rather than static, solidified clumps. That's a key distinction.
Precisely. This dynamic behavior immediately suggested a liquid-like nature, and a crucial observation was that the total fluorescent signal within these clusters remained nearly constant over time, which was critical.
Meaning they weren't being broken down.
Exactly. It suggested that these condensates were stable and not sites of degradation. This was a key piece of evidence that the ORPs were being preserved, not rapidly destroyed.
And how did you experimentally probe the biophysical properties of these condensates in live cells? Did you use any specific chemical tools?
We did, using hexaniols, which are short-chain alcohols known to disrupt certain biomolecular condensates by interfering with weak hydrophobic interactions. We tested 2 of them, 2-sulfide-hexanediol and 1-cumyl-6-hexanediol. 2,5,5-HD had no effect on Cis1 or new RPL26A localization.
Okay.
However, treatment with 1-pol-6-HD dramatically disrupted Cis1 perinucleolar localization, meaning Cis1 dispersed away from the nucleolar periphery, but it did not disrupt the RPL26A clustering.
Oh, that's interesting. Cis1 leaves, but the RPs stay clustered.
Exactly. This was a fascinating detail, suggesting that the specific biophysical interactions targeting Cis1 to the ORPs are distinct from the underlying interactions among the ORPs themselves that hold the condensate together. Remarkably, Cis1's localization was restored upon washout of the 1,6-HD, showing the reversibility of its association with this compartment.
That's a subtle but fascinating detail about the interactions at play. But to really dissect the biophysical properties of these condensates, you need more controlled conditions than a live cell. How did you manage to get around that and study them in isolation?
Right. This is where our ingenious cell-free lysate assay came into play, which proved to be incredibly powerful.
Yep.
We heat-shocked cells, then cryomilled them, effectively breaking them open without denaturing proteins, and resuspended the cellular components in a buffer designed to mimic the yeast cytoplasm. This allowed us to observe ORP condensates forming and behaving in vitro outside the complexity of a living cell.
So you basically created a miniature, heat-shocked cellular environment in a test tube, allowing you to manipulate specific factors, like cell biology in a dish almost.
Essentially, yes. We observed that new RPL26 and RPS4B formed distinct round condensates that co-localized with Cis1 mavenus, and these condensates were almost entirely absent in lysate from non-heat-shocked cells.
Confirming the heat shock dependence?
Yes, confirming that the formation was genuinely heat shock dependent. We even tried heating lysate from non-stressed cells to extreme temperatures, but still no condensates formed, indicating that the prior cellular heat shock history was essential for their formation, likely through some priming events. We also found that these in vitro condensates exhibited clear fusion and fission events, again visually confirming their liquid-like dynamics. Importantly, these condensates robustly excluded RNA, and adding RNAs to the lysate didn't disrupt them, strongly suggesting that the RPs within were truly orphaned from rRNA.
Okay, that cell-free system sounds incredibly powerful for dissecting the precise molecular mechanisms. What did it specifically tell you about the roles of ATP and, crucially, the major chaperone HSP70?
The role of ATP was quite clear. When we depleted ATP in the lysate using aporase, the round dynamic condensates were lost, and instead we observed the formation of much larger amorphous aggregates.
Aggregates, not nice round condensates.
Exactly. This immediately hinted at an energy-dependent process. Since Cis1 is known to activate ATP hydrolysis by HSP70, we then directly tested the effect of inhibiting HSP70.
And what was the outcome of HSP70 inhibition? Did it have a similar effect to ATP depletion?
Precisely. Adding the HSP70 inhibitor VER155008 effectively phenocopied ATP depletion. It resulted in the formation of large, irregular clumps of RPL26A. Critically, Cis1 still associated with these amorphous aggregates, which is consistent with Cis1 acting upstream of HSP70 to recruit it, but HSP70's activity was required to maintain the liquid state.
So Cis1 brings HSP70 in, but HSP70 needs to work using ATP to keep things fluid.
Exactly. We also found that while 1,6-HD could dissolve ORP condensates in the lysate, it failed to do so when HSP70 was inhibited, further underscoring the critical and active role of HSP70 activity in maintaining the condensate's properties and preventing solidification.
This strongly suggests HSP70 isn't just recruited, It's actively working to keep these condensates in a specific fluid state. To move beyond qualitative observation, how did you quantitatively measure this liquid-like property and the transition to a solid-like state? For our graduate student listeners, could you briefly explain the biophysical principles behind techniques like pixel variance analysis and bead tracking?
Absolutely. We use two sophisticated quantitative methods. First, the pixel variance analysis. A truly solid static sample or a perfectly homogeneous liquid will show a specific pattern of photon count variance equal to its average due to Poisson statistics. However, an inhomogeneous liquid, one that's dynamically rearranging through molecular movement and mixing, will show a higher than expected variance in fluorescence intensity over time within a given pixel. We quantified this quasi-normalized variance in RPL26A fluorescence over time. Pixels in untreated lysate showed a large variance, which is a clear signature of dynamic rearrangement and a liquid-like state.
More twinkling means more movement.
In a sense, yes, but critically, HSP70 inhibition significantly reduced this pixel variance, confirming a measurable transition to a less dynamic, more solid-like state.
So HSP70 is essentially stirring the condensate, keeping it fluid and preventing it from gelling up.
Exactly. It was a good way to think about it. To provide even more direct evidence of diffusion, we used a bead tracking protocol. We added fluorescent beads to the lysate and measured their mean squared displacement, or MSD, which is a standard measure of particle diffusion, within the condensates. HSP70 inhibition significantly reduced the MSD of these beads, indicating a dramatic 20-fold decrease in their effective diffusion coefficient, for instance, from 0.73 mimins in untreated lysate to 0.038 mimins when HSP70 was inhibited.
The 20-fold drop? That's huge.
It is. This was direct quantitative evidence of a transition from a liquid-like to a solid-like state when HSP70 activity was inhibited. And importantly, back in the cells, HSP70 itself, marked by CISO1, co-localized with CIS1 at the nuclear periphery during heat shock, strongly supporting its in-cell role in maintaining these dynamic structures.
This is truly where the chaperone-dispersed condensates part of the title really comes alive. The combination of live cell imaging and this ingenious cell-free lysate system allowed you to dissect both the macroscopic biophysical properties and the precise molecular players. What does this tell us about the fundamental active role of CIS1 and HSP70 in managing these orphan proteins?
It tells us something fundamental. The CIS1 HSP70 chaperone system is not just passively present. It actively maintains the liquid-like dynamics of these ORP condensates. Without HSP70's ATP-dependent activity, these otherwise adaptive reversible structures would quickly solidify into potentially toxic amorphous aggregates. CIS1 acts as the critical co-chaperone that recruits HSP70 to the ORPs, and HSP70's ATP-driven conformational cycle then acts to keep the components within the condensate in a fluid dynamic state. It's a sophisticated, active management system for crucial cellular resourcing under stress, preventing aggregation and preparing for recovery.
The idea of these condensates being liquid-like immediately brings up the question of reversibility. Are these ORPs simply stored away permanently, destined for eventual degradation, or can the cell actually get them back out and use them again when conditions improve? This is where the adaptive preservation aspect really comes to the forefront.
That was indeed the next big question, and the answer is a resounding yes. The observed liquid-like character of the ORP condensates in our lysate experiments hinted strongly at their reversibility in living cells. To test this directly, we pulse-labeled new RPL26A and RPS4B in yeast, subjected the cells to heat shock to induce condensate formation, and then allowed them to recover at a normal temperature of 30 degrees C, comparing this to cells undergoing sustained heat shock at 39 degrees.
Okay, the recovery experiment. And what happened to those condensates during recovery? Did they just melt away?
Pretty much. The results were striking. While the ORP condensates persisted and remained stable during sustained heat shock, in cells allowed to recover at 30 degrees C, the RPL26A and RPS4B signal rapidly dispersed from the nucleolar periphery back into the cytosol within a remarkably short time, approximately 7.5 minutes.
7.5 minutes to disperse. That's fast.
Very fast. Crucially, the total fluorescence signal remained constant throughout this process, indicating very little to no degradation of the ORPs. They were not being destroyed, they were being rapidly released back into the general cytoplasmic pool.
That's incredibly efficient. The cell basically hits pause on these valuable components, keeping them ready for action. Again, do you see the same phenomenon in human cells? Conservation is key.
Yes. The same relocalization pattern was observed in human HECT116 cells, pulse-labeled RPL26, dispersed from the nucleolar periphery back into the cytosol upon recovery from heat shock. This reinforces the conserved nature of this adaptive mechanism.
So they're released into the cytosol. But for adaptive preservation to truly mean something, these RPs need to be more than just release. They need to be functional again, incorporated into new active ribosomes. How did you provide that ultimate proof?
Right, this is where the story truly comes full circle, linking preservation to function. We performed polysome profiling, a standard biochemical technique that separates cellular lysates based on the sedimentation rate of ribosomes. This allows us to distinguish between free ribosomal subunits, monosomes, single ribosomes, and heavy polysomes, multiple ribosomes translating mRNA, which are indicative of active protein synthesis.
Okay, separating by ribosome load.
Exactly. As expected, heat-shocked cells showed a significant increase in monosomes and a decrease in polysomes, indicating a general slowdown in translation. But upon recovery, this was completely reversed, and normal, healthy polysome profiles returned.
So the translational machinery is back online, the factory reopens.
Exactly. And the most exciting part, during heat shock, the pulse-labeled RPL26A was found predominantly in the top layer fraction of the gradient, consistent with it being an ORP, not yet associated with ribosomes.
Free RPs at the top.
Right. But after recovery, that same pulse-labeled RPL26A shifted dramatically and sedimented in the heavy polysome fractions.
Down with the active ribosomes.
Precisely. This is direct and definitive evidence that the RPs orphaned at the nucleolar periphery during heat shock are indeed successfully incorporated into functional, actively translating ribosomes upon recovery. This, to me, is the core of adaptive preservation in action. The cell isn't just surviving stress, it's intelligently recycling its valuable components, preventing their degradation, and ensuring they're primed to hit the ground running once conditions improve.
So we've established that the ORP condensates are reversible and that the RPs are reused. But what's the specific role of CIS1 and HSP70 in this recovery process? Are they just passively holding the ORPs, or are they actively involved in their release and ensuring their functional reincorporation?
The CIS1 HSP70 chaperone system is absolutely essential not just for maintaining the liquid-like state, but also for the critical reversibility and subsequent utilization of these ORPs. In yeast, we used an anchor-away approach to conditionally deplete CIS1 from the nucleus. This allowed us to specifically control CIS1's presence during heat shock and recovery. When CIS1 was depleted and cells were subjected to heat shock and then allowed to recover, the RPs failed to disperse to the cytosol and instead remained aggregated adjacent to the nucleolus.
So without CIS1, that pause button for the ORPs gets stuck, leading to their solidification and they can't get out.
Precisely. And mirroring our earlier in vitro findings, lysosate from these CIS1-depleted cells showed that the ORPs formed amorphous, solid-like aggregates, again with significantly reduced pixel variance, indicating they had indeed lost their liquid-like dynamics in CIS1's absence. This directly links CIS1's presence and activity to both the biophysical properties and the physiological reversibility of the condensates. In human cells, inhibiting HSP70 activity also significantly impaired the export of RPL26 to the cytoplasm upon recovery, further confirming the conserved role of the HSP70 system. Both are needed.
Okay, so both CIS1 and HSP70 are critical for the release. What happens if these condensates do solidify? Say if the stress is prolonged or the chaperone system is compromised, are they then a lost cause, or can they ever be rescued? That's an important question, particularly if we think about disease states.
That's a critical question for both basic understanding and potential therapeutic applications. We performed an experiment where we transiently depleted CIS1 during heat shock, intentionally allowing the condensates to solidify.
Making them solid on purpose.
Yes, making them solid. And then released CIS1 back into the nucleus for recovery. CIS1 did re-enter the nucleus and co-localized with RPL26A, but initially the RPL26A remained condensed for over 105 minutes, much longer than usual.
Okay, so it took a lot longer to resolve.
Yes, but after about 120 minutes, the RPL26A signal eventually became cytosolic again, and crucially, without appreciable degradation.
So even after solidifying, the CIS1 HSP70 system can eventually resolve these stubborn aggregates, albeit much more slowly. That speaks to the sheer capacity of the chaperone system.
It does, demonstrating the impressive, albeit delayed, capacity of the chaperone system to resolve even stubborn aggregates. But this delay has significant consequences for the cell. We measured cellular growth rates during recovery from heat shock. Cells with CIS1 present and fully functional showed an immediate resumption of rapid exponential growth at a constant doubling rate. They bounced right back.
Okay, normal recovery.
In stark contrast, cells where CIS1 was transiently depleted during heat shock and therefore had solidified ORP condensates showed significantly slower growth and a much delayed plateau in their growth curve.
The functional relevance here for you, the graduate students listening, is profound. This isn't just an elegant biochemical mechanism observed in a test tube. It directly translates to how well a cell can bounce back from stress and continue to thrive. The ability of the Cis1-HSP70 system to keep those ORP condensates liquid-like and rapidly reversible is a direct determinant of cellular fitness and recovery kinetics.
Absolutely. It highlights a vital adaptive strategy. The cell could simply degrade these excess RPs, but that would be a significant waste of valuable resources, and resynthesizing them from scratch would take considerable time and energy during recovery. Instead, this system allows for a rapid return to full function once the immediate stress subsides, optimizing survival and recovery. It's smarter.
This research has some really broad implications, extending far beyond just ribosomal proteins. From your perspective as the lead author, what are some of the key conceptual shifts or larger takeaways for the broader field of cell biology that emerge from this work?
One of the core conceptual shifts, I believe, is reinforcing the adaptive role of biomolecular condensates, like stress granules, which store mRNAs during stress. These ORP condensates are not just passive accumulations of proteins, they play an active, adaptive role in the cellular stress response. Their potential benefits are multifaceted. They offer protection to aggregation-prone ORPs from misfolding and degradation, and they prevent their potentially toxic gain-of-function effects if they were allowed to aggregate uncontrollably. And as we just discussed, their reversibility confers a clear fitness advantage by enabling cells to rapidly resume proliferation after stress, effectively conserving valuable cellular resources.
It's active resource management, not just damage control.
Exactly. If we connect this to the bigger picture, this represents a critical evolutionary adaptation. Rather than simply destroying surplus components during stress, the cell has developed a sophisticated strategy to pause, store, and then efficiently redeploy vital resources, optimizing its long-term survival and recovery. It's a hallmark of cellular intelligence, if you will.
And this really shifts our view of the HSP70 chaperone system, doesn't it?
Yeah.
It's not just a protein folding and refolding machine, but truly a dynamic condensate remodeler, playing a much broader role in cellular organization during stress.
Precisely. This research suggests that while the initial sudden buildup of ORPs drives their condensation due to their inherent stickiness, it's the Cis1-HSP70 system that then actively intervenes to prevent them from transitioning into toxic static amorphous aggregates. This role of the HSP70 system in managing and dispersing condensates is actually quite broad, and we're seeing more and more evidence for it. It's been shown to actively disperse other stress-induced condensates, like those formed by PAB1, a poly-A-binding protein, and pyruvate kinase amyloids, and even to remodel transcriptional condensates formed by the HSR regulator HSF1 itself.
So HSP70 and its co-chaperones like Cis1 are really central to proteostasis, and a key, perhaps underappreciated, part of that role is actively regulating the biophysical properties and dynamics of these transient cellular compartments, keeping them functional and reversible.
That's a significant implication. We hypothesize that they may broadly surveil and remodel a wide array of biomolecular condensates during stress, making them central regulators of cellular organization and function, dictating how cells cope with and recover from challenging conditions.
This research obviously opens up a lot of new avenues for investigation. As a journal editor, one of the most exciting parts of reading a paper like this is the new questions it opens up. What are some of the key unanswered questions that your team is now pursuing, or that you see as critical next steps for the field, especially for our graduate student listeners who might be looking for new research directions?
Many fascinating questions indeed remain. For example, what is the precise relationship between ORP condensates and the nucleolus itself? The nucleolus is known to function not just in ribosome biogenesis, but also as a broader proteostasis compartment, sequestering misfolded proteins. So how do these ORP condensates at its periphery interact with its broader functions? Are there specific signals or structural elements that bridge these two roles? That's something we're very interested in.
The interplay between these different compartments.
Exactly. Biochemically, we still need to understand the exact mechanism of HSP70's action within these condensates. Is it acting primarily as a multiple turnover enzyme, constantly binding and releasing ORPs to disrupt their interactions and prevent solidification, or more as a holdase, blocking specific aggregation-prone regions on the ORPs?
The stir versus the shield model, perhaps.
Something like that. And how would you test the difference? We could imagine developing mutants that selectively affect one aspect of HSP70 activity over another, then seeing their effect on condensate dynamics and reversibility. That's a direction for future work.
And on the upstream side, how does heat shock actually inhibit RNA production so rapidly to initiate this whole process? You mentioned it's independent of HSR.
That's another important unresolved question. While it's known that RNA production is repressed during stress and that this inhibition leads to RRP accumulation and HSR activation, the exact molecular mechanism by which heat shock inhibits RNA transcription is not fully clear. There's speculation that perhaps the condensation or inactivation of key RNA biogenesis factors might be involved, leading to a quick shutdown of the RNA factory, but the details are still murky.
This raises an important question. For you, our listeners, what aspects of this research resonate most with your own work, or what new avenues of inquiry does it spark? The journey from understanding a cellular triage system to potentially finding new therapeutic targets for age-related diseases is truly incredible. And that leads us to perhaps the most profound implication of your work.
Indeed. The direct link we established between the biophysical properties of ORP, condensates specifically, whether they remain liquid-like or solidify, and overall cellular fitness is a powerful concept that extends beyond just ribosomal proteins. This has profound implications for understanding neurodegenerative diseases. We know there's an age-associated decline and loss of chaperone expression and function, particularly in critical tissues like the brain.
Right, the proteostasis network gets weaker with age.
Exactly. This loss of chaperone activity may very well lead to a decrease in condensate liquidity in neurons, causing adaptive, liquid-like biomolecular condensates to aberrantly transition into maladaptive solid aggregates, which are a hallmark of many neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS.
So the failure to keep these condensates fluid could be a direct driver of pathology in aging neurons.
That's the hypothesis. Understanding the mechanisms that maintain these adaptive condensates in a liquid-like state could actually offer new therapeutic strategies to prevent or even reverse disease progression.
That's a huge potential connection.
Absolutely. Identifying and understanding the molecular mechanisms that sustain the liquid-like properties of ORP condensates and biomolecular condensates in general could function more broadly to prevent and perhaps even be therapeutically harnessed to reverse the transformation of adaptive condensates into maladaptive toxic aggregates. It's a compelling link between fundamental cell biology, the aging process, and human health.
Well, this has been an incredibly insightful discussion. Today, we've taken a deep dive into how cells, under the immense pressure of heat shock, don't just discard valuable building blocks like ribosomal proteins. Instead, they intelligently sequester them in dynamic, chaperone-regulated condensates, ready for reuse when conditions improve. It's a testament to the cell's remarkable adaptive capacity and truly a highlight of contemporary cell biology research. Let's quickly recap the key takeaways for you, our graduate student listeners. First, we learned that orphan ribosomal proteins rapidly accumulate at the nucleolar periphery during heat shock. Second, the CIS1, or DNA JB6 in human cells chaperone system, is actively recruited by these ORPs. Third, CIS1, in concert with HSP70, actively maintains these ORPs in a dynamic, liquid-like condensate state. Crucial. Fourth, this liquid-like state is absolutely critical for the rapid and efficient redispersion of ORPs, allowing them to be incorporated into functional ribosomes upon stress recovery. And finally, the ability of CIS1 and HSP70 to maintain this condensate reversibility directly impacts cellular fitness and the cell's ability to quickly resume growth and function after stress. It's all connected.
And thinking forward, this work makes us consider a profound question. How many other cellular components, previously thought to be simply degraded or aggregated during stress, are actually being adaptively preserved in a similar chaperone-regulated manner? Could the widespread phenomenon of biomolecular condensation be a universal strategy for cellular resource management and adaptation? And if so, how many diseases stem directly from the breakdown of this intricate balance where liquid-like structures turn into solid pathological aggregates? That's a big question for the field.
The fantastic final thought to leave us with. We hope this deep dive has given you, our graduate student listeners, a fresh and inspiring perspective on protein quality control, cellular resilience, and the fascinating world of biomolecular condensates. Keep asking those big questions, keep digging into the details with rigorous methodology, and keep pushing the boundaries of what we understand about life's fundamental processes. Thank you for joining us for this deep dive, and a huge thank you to our guest, the author of this groundbreaking paper.
It was my pleasure. Thank you for having me. It was great to discuss it.
Until next time, keep exploring, keep questioning, and stay well informed.